Okay, well, hello everyone. My name is Virginie Sonnet, and thank you for coming for the last presentation. I did my master thesis at the Laboratory of Oceanography of Villefranche-sur-Mer with Lionel Guidi and Sakina Dorothea-Ayata. And I was mostly working on the morphological diversity of marine phytoplankton. So the main objective was to use imagery to look at the variation of morphology within the year and to see if there were or not periodicities linked to daily cycles, like tides or diel cycle linked to light. So as I said, I was working with phytoplankton. So phytoplankton has those amazing microscopic guys that have a lot of different shapes, as you can see on those pictures. And they perform photosynthesis. So they're important for taking up anthropogenic carbon from the atmosphere and also releasing oxygen. So actually, they release half of the oxygen that you breathe. They are taking part into biogeochemical cycles, so it's also important into modeling for future climate. And they're the first trophic level of the food webs. So they have high importance for fisheries. We were studying their morphology in a large estuary on the east coast of the United States called Narragansett Bay, in a small station here. And this estuary is, so as I said, between Boston and New York, and it's important both for tourism and for fisheries in the New England region. That's why water quality, and especially phytoplankton, has been monitored on a regular basis for several years. What we use to look at the morphology is an Imaging FlowCytobot. So it's an imagery device that combines flow cytometry and images. So we get pictures of individual cells or colonies. And then with a MATLAB process, we get the actual organism from the background, and we compute a lot of different morphological features, like size, perimeter, circularity, texture. We have more than 200 of those. So we decided to only focus on 1 year of data, because 1 year of data was already 77 million images, which was kind of hard to fit in R. So we decided to start with that and then expand later. And why did we decide to use morphology of phytoplankton and not just only taxonomy? Well, the ecological functions that I just mentioned about metabolism, predation, are often linked to morphology and especially to cell size. And morphology is the only characteristic that's easy, fast, reliable, and that we can get for all individuals. Because usually when we look at communities, we look at species. We're going to look at different nutrient uptakes for different species. But that can vary between organisms, between individual cells. And morphology is, like, so far as for today, one of the only ones that we can get for each individual cell. And this device that takes phytoplankton cells from 5 to 150 microns, it gives a sample each 20 minutes. So that's a very high frequency, and we can look into very short-term periodicities at the daily scale. So as I mentioned, the two objectives of my study were to look into the morphological diversity variations along the year in this shallow estuary, and to see if there were or not presence of periodicities related to tides and to the diel cycle of the light. So we did a 3-step analysis. We pre-processed the data, so we cleaned the images, we chose which morphological features we would use, and we subsampled, because 77 million images was still too much, so we took 100 images per sample. And our second step was dimension reduction, so that we could extract meaningful information from our data set. So we did a principal component analysis to have the main drivers of the variation, and we kept only the 4 first dimension. And on this morphological space that was created, we clustered the individuals in 150 groups using a K-means clustering. And for both of those analyses, we used a weight that was linked to the abundance recorded by the Imaging FlowCytobot. In order to have the finest details, we went back to our 76 million images after cleaning, and we reprojected them in the morphological space, and we assigned a cluster number to all of them so that we can have the big picture. The last step was the actual time series analysis. We looked at morphological succession just with trend analysis, graphical analysis. Morphological diversity was investigated with diversity indices, and periodicity with a wavelet analysis. Wavelet analysis is a very specific type of time series analysis that allows you to look at different periodicities, see if they are significant or not, and to look into time. So one periodicity might be significant the first half of the year, but not the other half of the year. So our results. First of all, we wanted to characterize the morphological space. So looking at the PCA, well, there are a lot of morphological features, but you can see that most of them on the right part of the first axis are linked to perimeter, area, major axis. So that's really the size and the length that are driving 40% of the variance. 20% of the variance is mostly driven by thickness. So a rotated bounding box, Y is actually the thickness, the minimum axis also, some are correlated, of course. And we can really see that if we plot in the morphological space images of organisms stacked on top of each other. So you can see that on the right, I hate this pointer, sorry. You can see that on the right you have more elongated and bigger organisms, while on top of the second axis you have thicker organisms. And eventually, the third and fourth axes are mostly driven by the regularity. So in yellow, for instance, you have the measures linked to symmetry, and you have also the complexity of the perimeter. And the fourth axis is driven by the number of blobs, which is actually how many organisms are counted on one picture, and it's a sign of coloniality. So as we've seen, so the main characteristic was size. So we decided to color our 150 groups from the clustering, from dark blue, the smallest on the left side of the PC1, to yellow, the biggest and longest, on the right side of the first axis of the PCA. And we plotted the proportion, I don't see the point, the proportion at each date along our year. So the white parts are gaps in the sampling because of maintenance. And what we can really see directly is that there are big peaks of bigger organisms, like more yellow. And those peaks are not smooth. That's not a smooth transition. It's really going to a community of bigger organisms and then coming back to a community of small organisms. Except for those peaks, no, sorry. Those peaks are also characterized by a very high diversity, functional diversity, in the sense that there is more richness, there is more diversity in it, so more resilient communities, actually, when we looked into diversity. And except for those peaks, most of the proportion of the organism is mostly small and medium small organisms, well, what I call subjectively medium small organisms. Those who are on the left side of the PCA with blue groups, and that we could relate to nanophytoplankton, for instance. And you have some examples of pictures here. If you want to go more into characterization of different groups, we can look into the other axis. So we can look into thickness, regularity, and number of blobs. And some specific peaks, for instance, can be related to phytoplankton groups. If we look into imagery, because we have imagery, so we have images, we saw that, for instance, in April, we have very thin and very long organisms. And actually, there were a lot of pictures of colonies of diatoms that are long and very thin, like Leptocylindrus danicus or Pseudonychia. Like in December, we did the same. We looked at the images, and we saw that when a lot of blobs are detected and the thickness is medium, still long organisms, it's a lot of skeletonema. And a third example, like in March, we have the same signal, but irregularities that decreases a lot compared to December, and that's because we have more and more Chetoceros. So we can do that for different parts of the time series, and what's interesting is that, for instance, organisms like skeletonema and skeletonema, they're the same species, but they have a very different morphology if they're in June or if they are in December. Same for Chetoceros in late March that are single cells, while most of the times they are found as colonies. And if you only do taxonomy, then you will just end up with them in the same category. While if you look at morphology, then you will be able to differentiate them, because they might have different properties for the ecosystem functioning. So that was the first part of my analysis. The second part was to look into periodicities. So here's the graph. I'm going to explain it, even if it looks complicated. So you have the time on the x-axis, and you have the periodicity on the y-axis. So it goes from 2 days periodicity to 32 days periodicity. And the colors stand for the power of the periodicity. So if you are yellow, that means that both the tidal range and the daily average size don't show a periodicity. If you are dark blue, as you can see here, for instance, there is a very significant periodicity both for the tidal range and for the daily average size. And the black lines actually contour which periods are found significant at a 10% level. It goes with the right graph, which is the average power, and which really allows you to see where are the main peaks of periodicities, where the periodicity is the most important all year long. So we can see that there is one peak at 27.5 days, and that this peak is present almost all year long, except in November and in May. And this peak actually corresponds to the perigee-apogee cycle of the moon. The perigee is when the moon is closest to the Earth, and the apogee is when it's the furthest from the Earth. When it's closest, that means the tides are more important, both lower tide and high tide. The second peak corresponds to the 15-day cycle, and it's the spring neap tide cycle that we're more accustomed to. And this one is not present all year long. It's mostly in May, late April, May, and June more. And what's interesting is when we look at phase difference, so when is the size the highest, and when is the tidal range the highest, and when are those peaks apart, we saw that there was mostly happening during low turbulence, intermediate turbulence periods, so after neap tide, when the turbulence is the lowest in terms of neap tide cycle, of course. So the second part of the periodicity was to look at the diel cycle, so with the time of the day, and this time the hourly average thickness, so that I can change from size. And you can really see that you have a 24-hour peak, and this one is significant all year long, because, well, you can see it. Oh, sorry. And we have a small 12-hour peak also, but it's significant only on smaller time scale, so we didn't really have time to investigate this one. And when we look at phase difference, well, since size was the most important characteristic, we were expecting phase difference with size to be kind of constant along the year, which is absolutely not the case. It's super highly variable. There is no pattern at the large scale. But on the contrary, we can find more regular and thicker organisms around dusk, around 8 p.m. approximately, almost all year long. And we can find more colonies around the middle of the day. We did the same analysis with the current speeds, that we can look at the tidal cycle during the day. And there was a very weak periodicity linked to 6 hours and 12-hour 30, which is the tidal cycle usually. And that was what we were expecting to be significant. So it wasn't significant at all. So to sum up, in terms of morphology, size is the main driver. And it goes along with thickness, regularity, and the number of cells. But we didn't find texture or opacity, for instance, to be important in the main axis. And in different analyses for different organisms, and also for different instruments, you can find texture to be important. So maybe the IFCB is kind of blurring this signal. The second findings that we had is those big peaks in the blooms of bigger organisms, that can be differentiated into different phytoplankton groups. And the importance of the nanophytoplankton, which is usually not really counted in taxonomic studies because it's very hard to identify or just put in one big group. And the advantage of having imagery is also the possibility to link it then further with taxonomy and to look at the images, actually. And for the periodicity, as I said, we were expecting a periodicity with high-low tide or vertical or horizontal movement of water, and we didn't find it. And we were expecting it because for chlorophyll at the same exact location, it's been demonstrated that it's anti-correlated with tide, so that chlorophyll is higher during the lowest turbulence, when the tide is at its lowest. On the contrary, but we found that still during low turbulence, intermediate turbulence, but at the month scale, there was a peak inside, especially in winter, spring. The blooms were happening almost always a few days after neap tide. And in the end, the 24-hour periodicity was the most significant, and we had, like I say, more colonies at midday and more regular thicker organisms at dusk. And the only solution, well, the only thing that could explain that we found in the literature is the cell cycle. because most of the phytoplankton cells in the literature are reported to divide at dusk. So that might explain why they're more regular and thicker before, but then we would also have that for size, and we don't have it. So it's kind of really strange. And as a perspectives, well, it was a first step in our analysis. We still have a lot of things to do. We are going to add light on nutrients that were not available during the analysis, and we want also to compare more deeper with the chlorophyll A so that we can have a better understanding so what's at the origin of those changes in morphology. We want to confirm it with a longer data set, of course, because we have, as you've seen, a lot of periods where we have missing data, and because there are seasonalities at 4, 6, 12 months that could also be investigated. And we could also look at very transient events. And in the end, what's going to be the purpose of my PhD is going to be combining the taxonomical approach, this morphological approach, and also the optical properties that are recorded at the same time, so that we can better understand the phytoplankton dynamics, which properties are linked to which phytoplankton groups, and hopefully, maybe, increase the accuracy of ocean color algorithms, especially in coastal waters. So I'd like to thank my two supervisors, Lionel Guidi and Saki Nayata, as well as Colin Mu and Audrey Ciochetto from the University of Rhode Island, who provided the data and helped me with the technical aspects. And thank you for listening for this last one.